This is our summary not only for DNA, but how we go from DNA to making protein. This is a section that traditionally students do really well on in terms of the test. So hopefully you're getting your confidence up and the test is going to go well. First part talks about the importance of DNA. It allows our genetic information to be passed to future daughter cells, and in many cases for organisms, future generations of organisms. And there's a lot of vocabulary to know. So you should know that chromosomes are made of DNA, and on the DNA are sections, specific sections of DNA, and they're called genes, and they code for a specific protein. So the first part of this game was understanding DNA structure. Double helix, a nucleotide made of a sugar, a phosphate, and nitrogenous base. The rails of the ladder, if we think of it as a ladder, is the sugar phosphate backbone. The rungs are the nitrogenous bases that give us the A, T, C, and G. And yes, you should know those words, but often we symbolize them with a capital letter. We should know that A and T are complementary, C and G are complementary, and that just means they fit together. Then we talked about DNA replication. So again, if you need more detail, go to specific notes, watch the video. But DNA replication means how does it make a copy of itself? And of course, this process happens in the nucleus where we find the chromosomes. So DNA unzips, nucleotides fill in a complementary fashion, always A with T, C with G, and the DNA zips up. And you should know why it's called semi-conservative. Then the rest of the section was on protein synthesis. It had two processes. Transcription, you see, occurs in the nucleus. The messenger RNA goes and gets information from the DNA. So the DNA will have unzipped, nucleotides fill in, creating the messenger RNA, which is a single strand. It contains uracil, the U instead of the thymine, the T, and because it's a single strand, it can come out of the nucleus and go to the ribosomes. So you have to know the process of transcription. Then, when you get the messenger RNA getting to the ribosome, translation can occur. The messenger RNA will feed through the ribosome, and the messenger RNA contains triplet codons, a sequence of 3 letters that are going to code for a certain amino acid. The match to that, the complementary match found on the anticodons of the transfer RNA, will then come in and attach to the messenger RNA's codons. Attached at the other end of the transfer RNA is an amino acid. And you use the codons chart to determine, hey, what amino acid are we getting from the messenger RNA? So you have this chart, and many students said they like this thing better, the wheel rather than the chart. And so if you had something that went, say, A, and then an A, and then a C, well, then you can see it's going to be asparagine, just like that. And you might just write down the first 3 letters of it. That would be fine. If you had something that was, I don't know, UAC, then it's going to be tyrosine, UAC, tyrosine. So make sure you know how to use that chart. Anyways, once you put amino acids together, they form a protein. And you might say, what's this polypeptide? Well, it means you put amino acids together. Poly means more than one, so you put more than one amino acid together with something called a peptide bond. You should know the difference between DNA and RNA. DNA, double helix. RNA, single strand. Deoxyribonucleic acid contains deoxyribosugar. Ribonucleic acid contains ribose. The deoxy just means it's removed an oxygen atom. We should know that DNA is bigger than RNA. We should know that DNA cannot pass through the nuclear membrane, but RNA can. We should know that these are the 4 nitrogenous bases in DNA, and these are the 4 nitrogenous bases in RNA, and the difference is the T and the U. You should also memorize the name and function of different proteins. So we have hemoglobin, very important in oxygen for transporting red blood cells, and it combines with iron. Insulin is going to help cells absorb sugar, and so you don't get high blood sugar levels, you're getting it in the cells so it can be used. Keratin, hair and nails, and we carry on. Enzymes, hugely important for the chemical reactions in your body. Antibodies, things that are part of your immune system to help protect the body against foreign invaders. Fibrinogen, helping clot the blood. And lactase digesting milk sugar. And we carry on with that. So hopefully you've been studying on a regular basis. Hopefully you've had a chance to ask questions so you can make corrections if you've made mistakes in assignments. But it's up to you to study daily and make sure you're ready to dominate this test. And if you've forgotten, this test is on DNA and protein synthesis.